What is DNA transcription? The first crucial step in gene expression. DNA transcription is the process where a gene's DNA sequence is copied to create an RNA molecule. During transcription, the genetic information stored in DNA is read and transcribed into a new RNA strand. One key difference is that RNA uses uracil, or U, instead of thymine, which is found in DNA. Think of DNA transcription as making a photocopy of a specific page in a huge instruction manual. The DNA contains all the instructions for building and maintaining an organism, but only specific sections need to be accessed at any given time. Transcription creates a working copy, the RNA, of just the needed information, leaving the original DNA intact. Transcription is the first essential step in gene expression, where the information in genes is used to create functional products like proteins. The process begins with DNA, which contains the genetic instructions. Through transcription, this information is copied into RNA. Later, this RNA will be used to create proteins in a process called translation. Transcription is the critical first step that allows cells to access and use the genetic information stored in DNA. RNA polymerase is the main enzyme responsible for transcription. It travels along the DNA strand, reading the genetic code. As it moves, RNA polymerase builds a complementary RNA strand, matching each DNA nucleotide with its RNA counterpart. The resulting messenger RNA carries the instructions needed to build proteins in the cell. In summary, DNA transcription is the crucial first step in gene expression, allowing cells to access genetic information through RNA creation. Recent research has revealed fascinating new details about the transcription process. Scientists have captured in real time how RNA polymerase opens the DNA double helix to form what's called a transcription bubble. This discovery provides crucial insights into how transcription initiates at the molecular level. A newly discovered code within DNA, termed spatial grammar, influences gene activity based on the precise spacing between transcription factors. The spacing between transcription factors and their position relative to where a gene's transcription begins determines the level of gene activity. This spatial organization works like a language grammar, with rules that determine how strongly a gene is expressed. Recent studies have shown that ribosomes can attach to the RNA during transcription, creating what we call transcription-translation coupling. A coupled ribosome can increase transcription speed and processivity, though this often comes at the expense of accuracy in RNA synthesis. This means the ribosome isn't just translating the RNA into protein, but also acting as a transcriptional regulator that helps control the process on protein coding sequences. These discoveries are fundamentally changing our understanding of gene expression and the complex molecular interactions that regulate it. Transcription factors are specialized proteins that play a crucial role in regulating gene expression. These proteins must navigate the complex organization of DNA in the nucleus, where DNA is tightly packed into a structure called chromatin. Transcription factors can recognize specific DNA sequences, even when the DNA is wrapped around histones in nucleosomes. Once bound to DNA, transcription factors recruit RNA polymerase to initiate transcription at the promoter region. Recent studies are using single molecule technologies to observe how transcription factors interact with RNA polymerase and chromatin. These advanced techniques allow scientists to measure binding rates, cooperativity between factors, and the dynamics of transcription complex formation. Understanding how transcription factors interact with DNA packaging provides key insights into gene regulation. These proteins navigate complex chromatin structures, often working cooperatively to enable gene activation. The study of DNA transcription is driving advancements in several fields, opening new doors for applications and technological innovations. The DNA and gene chip market is expected to grow significantly from $8.9 billion in 2023 to $15.35 billion by 2029. Even more impressive, the global DNA sequencing market is projected to reach $51.31 billion by 2034, with a compound annual growth rate of 14.9%. These significant investments are fueling several key areas. Precision medicine is advancing through personalized therapeutics based on individual genetic profiles. Advancements in DNA sequencing technologies are expanding their use in clinical diagnosis, allowing for more accurate and rapid identification of diseases. A deeper understanding of transcription is enabling novel genome engineering strategies. CRISPR-Cas systems continue to evolve, offering more precise genetic modifications with applications in therapy and agriculture. Single-cell sequencing approaches are revolutionizing how we study chromatin and gene expression, allowing researchers to identify heterogeneity and understand cellular processes with unprecedented detail. Spatial transcriptomics is emerging as a powerful tool to map gene expression while preserving spatial information, revealing tissue organization and cell-to-cell interactions. Nanopore sequencing technologies are enabling real-time, direct RNA sequencing with long-read capabilities, making DNA and RNA analysis more accessible and field-deployable. The advancements in transcription research are having wide-ranging impacts across multiple sectors. From infectious disease management to cancer therapeutics, genetic disorder treatments, drug discovery, agricultural advancements, and microbiome research. As transcription research continues to advance, it will drive innovation across precision medicine, technology development, clinical applications, data analytics, and therapeutic advances. The projected market growth reflects the increasing importance and vast potential of this field to transform healthcare, agriculture, and our understanding of the fundamental processes of life.